We are so glad you are here with us today. I'm Ellen Herman, co-director of the Wayne Moore Center for Law and Politics and professor of history here at the University of Oregon. The mission of the Wayne Moore Center is to promote civic engagement, inclusive democracy, and justice by bringing together students, scholars, activists, policymakers, and members of communities, people just like you. We discuss a wide range of issues affecting Oregon, our nation, and the world. We work in the tradition of Oregon Senator Wayne Morse, who believed that political independence and education were building blocks of justice and democracy. This panel on reproduction and genetic technologies is part of the center's Science, Policy, and the Public theme of inquiry. If you aren't already on our mailing list and would like to hear about other events like this one, please sign up for our mailing list on our website. There's a field right at the bottom of the homepage where you can type in your email address. Just a quick couple of thank yous. Thank you to the amazing members of the Wayne Moore Center team, Thea Croman, Rebecca Dinwoody Flynn, Abby Stilley, Dan Titchener, and Christine Waite. They make our work a pleasure every single day. We are also grateful for support for this event that comes through the Lorwin Lectureship on Civil Rights and Civil Liberties. And we are especially grateful to the many members of the Wayne Morse Circle who are here with us today. Circle members provide critical support for our programs, including public programs like this one, through annual donations in any amount, small or large. Please consider supporting our work by becoming a Wayne Morse Circle member. We are celebrating our 20th anniversary this year, and all of us look forward to bringing you programs like this for at least another 20 years. And now I'd like to introduce my colleague, Judith Eisen, Professor of Biology and Distinguished Scholar at the Wayne Moore Center this year. Professor Eisen will introduce today's panelists.
Thank you, Professor Herman. I want to add my thanks to everyone for attending this event. Let me tell you a little bit about how things are going to go. So first, we're going to hear from Paul Knoepfler. Professor Knoepfler is Professor of Cell Biology and Human Anatomy at the University of California, Davis School of Medicine. He's interested in epigenetics and in stem cells and cancer. His lab uses cutting-edge methods, including genomic and gene editing technologies, to answer research questions with the goal of developing novel clinical interventions to benefit human health. He is the author of a blog, The Niche, that is widely considered the most important educational resource about stem cells and regenerative medicine. Its goal is to provide clear information about innovations in biomedical research, including stem cells and gene editing. Professor Knoepfler has also authored or co-authored several books for general audiences, including Stem Cells, An Insider's Guide, GMO Sapiens, The Life-Changing Science of Designer Babies, and most recently, How to Build a Dragon or Die Trying, a satirical look at cutting-edge science. Our second speaker will be Camisha Russell. Professor Russell is Assistant Professor of Philosophy at the University of Oregon. Her work in bioethics intersects with the philosophy of race, African-American philosophy, and feminist theory. She has written about transracialism and gay men and reproductive justice, among other topics. In 2018, she published The Assisted Reproduction of Race, which explores how salient race is in how people use reproductive technologies, which might seem surprising because of the scientific consensus that race has no genetic basis. Professor Russell is currently co-editor of Hypatia, a journal of feminist philosophy. She was recently awarded the 2021 Baruch A. Brody Award and Lecture in Bioethics from Baylor Medicine Center for Medical Ethics and Health Policy, Houston Methodist Hospital, and the Rice University Department of Philosophy. Our final speaker today will be our commentator, Françoise Baylis. Professor Baylis is the 2021 Wayne Morse Chair of Law and Politics. She's a philosopher and university research professor at Dalhousie University in Nova Scotia, Canada. She's known for her research and writing on assisted reproduction and women's health. Her most recent book is Altered Inheritance, CRISPR and the Ethics of Human Genome Editing. Professor Baylis is a member of the Order of Canada and the Order of Nova Scotia. She is a fellow of the Royal Society of Canada and a fellow of the Canadian Academy of Health Sciences. In 2017, she was awarded the Canadian Bioethics Society Lifetime Achievement Award. Professor Baylis is currently a member of the World Health Organization Expert Advisory Committee on developing global standards for governance and oversight of human genome editing. After our three speakers have finished, we'll field questions from you, members of our audience. We really welcome and look forward to your questions. We anticipate having plenty of time for these questions, so please use the Q&A box, which appears at the bottom right-hand corner of your screen, to submit your questions for our speakers. So without further ado, I'm going to turn this over to Professor Knoepfler.
All right, hopefully you guys can hear me well and see my screen. Does it look okay? So thank you for the introduction. It's really a pleasure to speak with you about something that's been of growing interest to me over the last 5 or so years. So this is just an introductory slide with a couple of resources about with links to my lab's own page about our research. And then as was said, I also have a blog and resource site that is important in terms of stem cell ethics and policy work that I've done. So I thought I would start out by introducing you to CRISPR-Cas9 gene editing. This is, of course, an explosive kind of technology in terms of culture and society. Recently, 2 Nobel Prizes were awarded for this technology. It's been on the cover of many popular magazines. So it's really sort of taken the world by storm. So I thought I'd give you a few minutes just to kind of make sure we're all on the same page. Some of you may know a lot about CRISPR, and others it might be a little bit of a newer area for you. So interestingly, as much as now CRISPR has sort of taken humanity by storm, the story really starts back in primordial war, if you will, between bacteria and viruses. So for thousands of years, bacteria have been infected by certain kinds of viruses, and bacteria have evolved a way to fight back by essentially nuking the genome of viruses that attack it with something called nucleases that chew up the virus's genomes and basically kill them. Clever scientists figured out that they could adapt this technology for our use to make very precise changes in the genome of cells. And as many of you may know, there are 4 units in DNA, A, C, and G, and T. So with CRISPR-Cas9, you can change as little as 1 A to say a G or a T. So it really allows you to kind of recode the DNA of different kinds of cells, including those of ourselves, human cells. I've been a genetics researcher for a few decades now. I guess that kind of shows how old I am, but it also allows me to kind of compare this new technology to older technologies. And CRISPR, there's a reason it's become so somewhat almost like an obsession amongst genetic researchers and culture in general. Compared to the older technologies I sort of grew up on as a scientist, CRISPR is far faster. We're talking about months rather than years. It's far cheaper. We're talking about hundreds rather than probably tens of thousands of dollars for doing CRISPR research. It's also generally much more efficient than the old-fashioned techniques, again, I used as like a grad student. And it also may leave no mark in the genome. So like I was saying, if you want to change an A to a T or something like that, with the old-fashioned techniques, you'd also often sort of leave sort of a scar in the genome behind along with that change you wanted to make. CRISPR doesn't generally do that. CRISPR is also more specific than the older techniques. And the parentheses there, the not perfect part, is really important, as you'll see for the rest of my talk, because sometimes I think CRISPR is portrayed as almost perfect. So for us, it's interesting, like my lab does research on mice, and we also use human cells. Other labs study, worms and flies. And one of the most striking things about CRISPR is it works so well in our own cells and in human cells. And this includes stem cells and other kinds of cells, including germ cells. It even works in human embryos, as I'll talk about more. So this obviously opens the door to something well beyond just research, right? If this technology can actually make genetic changes in human embryos. So as I mentioned, for some of these different reasons, this has become ubiquitous in science, and also taken sort of pop culture by storm in some ways too. I think I even saw a movie a couple of years back about CRISPR creating these giant Godzilla-like monsters or something like that. So CRISPR can goof up too, though. I mentioned it's not perfect, so it can kind of throw a monkey wrench into the genome of the cells that you're trying to make a specific change in. And so there are a couple different main ways this can happen, and this kind of helps explain how CRISPR works. So normally, let's say we want to target a gene, we'll just call it gene A with CRISPR-Cas9, and let's say that gene has a mutation in it in these cells, and that mutation leads to a human genetic disease. So we want to target gene A and change the mutation back to a quote unquote wild type or normal state. The way CRISPR does this is actually involves a nuclease I mentioned earlier. So you end up cutting the DNA, cells try to fix that cut. And in that process, it kind of gives us an opportunity to recode the DNA. So as part of that system, CRISPR has kind of a GPS-like system where it can find a precise location in the genome. The genome is kind of this big universe unto itself of billions of these DNA units called base pairs. So sometimes one thing that can go wrong with CRISPR is, let's say, again, we're trying to target gene A. Oops, CRISPR might go to the wrong place. And we call this an off-target effect. So it might cause a genetic change in gene B, C, or gene Z, some other gene, and this could have catastrophic effects. We've also been learning in just the last couple of years how often CRISPR sometimes can go to the right place, but still really screw things up. So again, let's say we're trying to make a mutation in our favorite gene, gene A. CRISPR will sometimes go there, so that's good, right? But it will do the wrong thing. So instead of changing an A to a C or something bigger, some kind of very precise but a little bit bigger change, it can make a different mutation. And that, of course, can be a bad thing. Different mutations can cause different kinds of effects. So that's definitely not a good thing. We've also learned that CRISPR sometimes can lead to big deletions, let's say, in a gene. So let's say we're trying again to make a mutation in our favorite gene, gene A. Sometimes CRISPR will just lead to that entire gene getting lost. And this third bullet point, we're now realizing sometimes the entire chromosome that gene is on sometimes can be lost, which is obviously a huge deal. Chromosomes are important, right? Cells like to have all their chromosomes. Things can really go wrong if a cell loses an entire chromosome. So, these are things we're having to grapple with as we're doing CRISPR research, like I'm doing it on stem cells and cancer cells in my lab. If we're trying to model a mutation that causes cancer, but we lose an entire chromosome, that's a big problem for us in terms of our research. What makes it even more complicated is that these kinds of negative outcomes are, although we're aware of them and we're more aware of them now than we used to be, they're kind of hard to predict. So we can use CRISPR computer algorithms to help us kind of guide our work and predict might go wrong, but sometimes things go wrong that we're not predicted. The other thing that really can be a headache is, let's say we're trying to predict what can go wrong and we think maybe something did go wrong. Again, the genome is a huge place as much as it's this tiny little thing. So it's hard to kind of navigate through. Imagine trying to find one restaurant in New York City or something like that where something has gone wrong that you need to go there. So detecting the glitches can be a big problem. The glitches also probably depend on the gene you're trying to gene edit. They probably depend on the patient. So for all of these reasons, some of us has sort of started raising the question of whether or not, when we're talking about CRISPR-Cas9, is gene editing even like the best name for this? Because oftentimes, gene editing, to me at least, implies a precision, right? Like you're going in there, you think of like an editor. It also implies making something better in some way, I would think. Like if you're editing, hopefully your editor of like a manuscript or paper is not making things worse, but making things better. So there's also been some debate, is the idea, the name gene editing sort of giving the public the wrong idea about this technology. And especially when it's, CRISPR-Cas9 is not making a precise change, but let's say you're trying to use it for a research purpose to get rid of an entire gene or something like that. That's a huge chunk of DNA you're kind of cutting out. And sometimes that's pretty random. And so, is editing really the best metaphor? So I think in some ways what's generated the most attention, or a lot of attention, as compared to some of the things like in my lab, we're just studying cancer, but what's generated more attention than just sort of some of the basic or translational research is the idea, again, that we can put CRISPR-Cas9 into human embryos. And while some labs have already done that, you could, instead of studying those embryos in a lab, you could implant them in a surrogate mother and create new people, right? And if you put CRISPR-Cas9 in there, you could change things up in a way that is not so simple. So the appeal, I think, of the idea of going germline, what's also known as heritable CRISPR in people, is that you could, in theory, eliminate genetic diseases forever in certain families. That's a really powerful idea. These genetic diseases can be just horrible and decimate families. And what I've learned over the last 5 years that I didn't really, wasn't so much on my radar screen before that, is that there are already existing technologies where you don't have to try to edit. You can just screen embryos instead at IVF or fertility clinics. And one of the names for this kind of technology is called PGD, or pre-implantation genetic diagnosis, where you pluck a few cells from an embryo that was made by in vitro fertilization. And you can analyze those and you can say, oh, these embryos have a mutation that causes Huntington's disease, but these other embryos from the same couple don't. So using this technology, they implant the embryos that were found by screening to not have mutations, and then you get healthy births. So it seems to me like one of my feelings is that screening is a much better way to go than to try to CRISPR embryos, given all the things that CRISPR can cause to go wrong. There are also technical issues related to trying to use CRISPR in a reproductive kind of sense that make it really difficult, much more difficult than like in my lab, we can CRISPR 1 million cells and just take our time analyzing them. But if you're doing this with a human embryo, you don't really have that luxury of the time to do validation. So some of the issues, in fact, may be so difficult, we can't really resolve them. Even the sort of advocates of trying heritable CRISPR in humans acknowledge some of the things that can go wrong. There's just no way to easily test for them. And one of these things is called mosaicism. And what that means is that cells in one organism have different DNA. So, we all were taught that all of our cells in our body have the same DNA. We might actually have slight differences in DNA in our different cells, but not to a big extent. But if we try to use CRISPR in embryos and kind of put it in there, like say in a fertilized egg in a fertility clinic, you can end up with an embryo and then a person, should you take the chance of doing this in a reproductive kind of way, where like 10% of the cells might have one kind of DNA, 50% might have a slightly different kind of DNA. That's the kind of mosaicism, and that kind of mosaicism from CRISPR could lead to diseases and other really serious issues. So for me, as a scientist, I often think a lot about these sort of technical issues, but obviously there are other issues that are really important here and things that we need to think about and address, and we need to talk about them way more than we already have as a society and as different cultures around the world. Using CRISPR in a heritable way raises many ethical and societal, social justice kind of issues. I've also realized over the years there are many different cultural views about making genetic changes in people, and this varies a lot around the world. Another issue is that I think, having thought very carefully about this, there isn't a real bright line between using CRISPR to try to prevent a genetic disease and to try to use it maybe, say, to change a trait, like make someone who has bigger muscles or some other kind of change. There's not as much of a line there between these things as you might think. So it's been interesting for me. I've been kind of following this over the last 5 or 6 years, both as a scientist whose lab is using CRISPR, but then also more on the policy and ethics side. And one of the things I thought was really interesting, a number of years back, there was actually an anti-CRISPR protest in France where people were walking around with signs with these logos I'm showing here, basically like no GMO kind of baby, no, no CRISPR. And it does seem to be the case that in different parts of the world, especially like in Europe, there are much stronger feelings about not using CRISPR than say in the US, where it seems like more of a mixed bag, at least in terms of heritable use of CRISPR. I also had this really interesting experience of giving a TED Talk in Vienna in 2015 about CRISPR and the potential for designer babies. And one of the things that really struck me about that whole experience was just, I met with so many different Viennese people. There were also people from other European countries there for that event. And it was basically almost uniformly strong opposition to just even the idea of using heritable CRISPR in people. So that was a very sort of strong personal experience for me to realize, how different those feelings were than say within my own scientific community of researchers, where some people actually seem a little bit more inclined to leave the door open for that. Another thing is, as we think about using CRISPR in people, what would you even do? Let's say we're not necessarily going to try to prevent diseases, but we're going to try to make some other kind of change in people using CRISPR in a heritable way. What would you even target? What genes would you try to change? And interestingly, there already have been discussions about this out there in the world. And one of the proponents of kind of leaving the door open has been this Harvard geneticist, George Church. And he even came up with a list of 10 different genes that he might think could be reasonable to try to CRISPR in people, either in a non-heritable way or even in a heritable way. And you can see the associated traits here on the right, that some of these are really striking to think about. Would you really go to the trouble of trying to CRISPR a person, give them a permanent genetic change just to change like their body odor or other things? So this list really always struck me as quite remarkable. And I wanted to highlight two particular genes on the list, one called myostatin, or MSTN, that is kind of a gene that sort of modulates our muscle development so that we don't get muscles that are too big in a sense. And then this other gene called CCR5, that's a cellular receptor that HIV in part uses to enter the cell. So both of these have been talked about a lot in terms of potentially CRISPRing in people. And I just wanted to show you, hopefully you guys can see this picture of these pigs. And the pig in the upper right inset is a normal pig. And then these other pigs down here are sort of the Arnold Schwarzeneggers, of the porcine world. They had their myostatin genes CRISPRed, and they ended up being sort of these giants. And there were a number of sort of unintended consequences of this, including extremely difficult deliveries for the sows who were pregnant with these genetically modified or CRISPRed pigs. So the other gene, CCR5, has really also gotten a lot of attention. And many of you may know that a couple of years back, a researcher in China CRISPRed this gene, CCR5, he attempted to at least, in actual human embryos and implanted them. And as far as we know, generated 3 children who have CRISPR gene edits. So these were sort of the first CRISPR babies, first people with heritable genetic changes due to gene editing. It was a whole fiasco, I think, in many ways. And the researcher He Jiankui, as far as I know, is currently serving a prison sentence in China for various issues related to how he conducted this research. The children themselves, we really have very little information, in part due to privacy rules. But as best as we can know or kind of decipher from a little bit of data he presented at a conference and then some other data that kind of came out, the gene editing went pretty badly that he was trying to do. He kind of messed up that CCR5 gene. He also got some of that mosaicism that I was talking about, most likely. So ultimately, the guess is that these children are not actually in any way protected from HIV, and they may have other risks from basically genomic damage from CRISPR that at this point we don't really understand. So it's really a concern about the future for those children. So to wrap up, I just want to emphasize how much potential I think CRISPR has for laboratory research. There are different things I didn't have time to talk about, like disease modeling, drug screening for different genetic diseases. There's also been a lot of excitement about somatic uses, non-heritable uses of CRISPR in people, such as for sickle cell disease. But again, it seems like there's sort of this gravity pulling us back towards heritable use of CRISPR in people. But there again, there's so many technical issues. There are other societal issues. I think unfortunately, there's still a lot of enthusiasm among some researchers to push ahead with this in the future. And I just wanted to add a note that as much as putting CRISPR into fertilized eggs has a number of challenges, the way things are trending right now, it seems more likely that in the future, researchers will try to gene edit some special kind of stem cells called germ cells that you can then use to make sperm or egg and then use for IVF to try to make people. So that may be sort of the technical future for that kind of approach for heritable gene editing. So I'll stop there and can move on to the next speaker.
Okay, thank you, Paul. I am the next speaker. So let me get my slides up here. Okay. So, whoops, let's go back. Nope. There we go. Okay, so as I say on this slide, I'm going to give some thoughts on the relationship between race and eugenics. And so I'm thinking throughout this presentation about genetic technologies as a form of eugenics, right? Because they're trying to improve human beings through genetics in some way. So I'm just going to start with 3 quotes that help to kind of get at what I'm getting at. So we're making a shift towards humanities here in this panel now. So the first quote, our race is overweighted and appears likely to be drudged into degeneracy by demands that exceed its powers. We can, in some degree, raise the nature of man to a level with the new conditions imposed upon his existence, and we can also, in some degree, modify the conditions to suit his nature. It is clearly right that both these powers should be exerted with the view of bringing his nature and the conditions of his existence into as close harmony as possible. So then the next one, it is significant that we have reached a point in human history at which further attempts to make the world a better place will have to include not only changes to the world, but also changes to humanity, perhaps with the consequence that we, or our descendants, will cease to be human in the sense in which we now understand that idea. And then the third and last quote, as you probably are only beginning to realize today, in times when genetic screening, testing, and patenting pervade all sectors of social and economic life, and with the synthetic powers of genomics on the horizon, the epistemic space that heredity came to constitute has reconfigured life in its entirety. So these 3 quotations represent the wider discourses that I'm hoping you can think about together during these remarks. The first quote is from Francis Galton, the man who coined the now infamous term eugenics in 1883. And it represents the discourses of the original modern era eugenics movements. Though diverse, these eugenics discourses of the late 19th and early 20th centuries were permeated by the language of race. They were also deeply nationalist discourses, concerned with maintaining or recovering not only biological, but cultural vigor. The cures they offered for the perceived ills of modern or Western civilization were based on an emerging understanding of the scientific principles of heredity. The second quote represents the discourses of contemporary so-called liberal eugenics. The language of race has no place in these discourses except as part of a disavowal of the racism of the bad old eugenics. They're deeply neoliberal, concerned with our personal responsibility for broader societal or global well-being as exercised through our free personal choice. They propose solutions to the perceived threats of global environmental disaster or human extinction based in the contemporary science and technologies of genetics. In other words, liberal eugenicists wish to reclaim the word eugenics, protecting its practices from past injustices by ensuring that all genetic technologies are freely chosen by individuals only for use on themselves and or their children, or in the case of heritable genetic editing, their descendants in perpetuity. Finally, the third quote suggests that despite the effort of new eugenicists to distance themselves and their aims from the bad reputation of earlier involuntary eugenic and genocidal practices, the two sets of discourses exist in a shared and dominant epistemic space centered around the idea of an obsession with heredity. An epistemic space is a broadly shared way of knowing and making sense of the world that is so pervasive as to often go unnoticed or uninterrogated. As Troy Duster puts it, we have come to see the world through a prism of heritability. This epistemic space, I would argue, is also the home of the race idea and of scientific racism. So in these remarks, I want to engage in a very brief exploration of the shared epistemic space of heredity, race, and eugenics with the aim of addressing the larger question, can we have a eugenic project free from or purged of racism? To answer yes, I claim, would require understandings of race and racism that are both narrow and ahistorical. By contrast, a broader understanding of these ideas and of their historical context makes non-racist and truly just eugenics appear much more elusive. So the specific questions I want to address here are, what do eugenicists have in common? How is that related to the idea of race? How do liberal eugenics coerce? And why does race still matter here? So what do eugenicists have in common? Sometimes the association of eugenics with Nazi Germany can prevent us from either recognizing that eugenics was truly, prevent us from recognizing that eugenics was a truly transnational modernist philosophy. So far from being a unified movement with a single objective, eugenics at the turn of the last century must be understood as a broad coalition of persons or groups promoting overlapping yet diverse scientific, social, or political agendas, including in America alone, scientists, socialists, sexual reformers, immigrant radicals, physicians, agriculturalists, and popularizers. Among the differences to be found between various proponents of eugenics were whether a eugenicist favored positive eugenics, encouraging the most fit to marry amongst themselves and have large families, or negative eugenics, curbing the fertility of those judged least fit. And I think that genetic technologies we're talking about now are the sort of descendants of this positive eugenics. The extent to which eugenic interests were, number two, the extent to which eugenic interests were combined with a focus on race. Number three, the specific policies that were recommended. And four, political orientation. Of course, the fact of this diversity also points to just how common eugenic thinking was at the time. Moreover, even where eugenics language appears racially neutral or simply to be referring to the human race, the reality is that most eugenicists had in mind the improvement of the white race or races. So there were ideas about a variety of white races that we don't have as much anymore. Working on the assumption that other inferior races, the non-white races, were likely to degenerate and die off on their own. So in their ambitious jointly written work on questions of ethics and justice in the application of new genetic technologies to human beings, bioethicists Buchanan, Brock, Daniels, and Wickler identify three core tenets shared by most eugenicists of the early 20th century. The first was a concern about degeneration, understood either as a consequence of modern social processes interfering with natural selection by rescuing and nurturing the unfit, or as the result of race mixing. The second was a belief in the heritability of a variety of not merely physical, but behavioral traits like talents, temperaments, proclivities, and dispositions. Therefore, third was the belief that social problems had both a biological basis and, to some degree, a potential biological remedy, such that reproduction was seen by all eugenicists as an act with social consequences rather than a private matter. Buchanan et al. Go on to discuss what was and was not wrong about past eugenics. They do not think it is wrong for parents to avoid bringing into the world children with severe disabilities. In fact, they think it might be a parental duty. They also argue that most of the values pursued by eugenicists, like intelligence and self-control, are widely shared by the population. I don't know where they stand on body odor. They find, and if I had more time, I would take very detailed issue with this, that reproductive freedoms are sufficiently well established that we need not entertain serious fears about the return of coercive eugenics in the wake of the Human Genome Project. And so I just want to say that reproductive freedoms are not considered very well established by a lot of women of color, for example. So what did they think was actually wrong with past eugenics? It was the pseudoscience on which it was based, and the bigotry, racism, and class prejudices with which it was infused. So on their account, the eugenics movement was, in this respect, just a creature of its time. So they said, we should probably be glad that early eugenicists didn't have the genetic knowledge to actually change the human gene pool, because they wouldn't have had the wisdom to do it correctly. But working from the assumption that we now have the scientific and technological means to make a real difference in people's genetic lives, or that we're developing those means, their question then becomes how to do so justly. So in other words, they're arguing for a conceptual and ethical distinction between racist ideology and the idea of improving the health of a population through eugenic measures. They believe there are ways to develop genetic science and to deploy genetic technologies to pursue both private and public goods without bringing about the coercion and abuse that the term eugenics now connotes. Such arguments can be very compelling, and they seem to be based on solid reasoning, but I believe that they actually operate only on the surface of this issue. So how is this related to the idea of race? So I think that rather than seeing eugenics as the political misapplication of a sort of flawed racial science, we might instead see an essential human drive to master nature as that which underlies the epistemic space of heredity, which includes not only eugenics, but both the concept of race and the subsequent attempts to justify race scientifically. Eric Vogelin writes, modern race theories give the impression of aggressive optimism because, like technology, they are eager to put to use the lawful course of nature in order to arrive at a specific objective they consider desirable. So as the concept of race developed prior to the 20th century, race came to be seen as a force or cause in nature, and therefore as something to be mastered for optimal societal efficiency and yield. The relationship between the drive to mastery and race can be seen in the way that animal breeding as a technology provided the conceptual framework within which the scientific race concept emerged and developed, a fact which shows that eugenic thinking was much more essential to the race concept itself than is typically recognized. Indeed, one account of the origin of the term race traces its first emergence to the Spanish language, where raza was extended to human beings from a more primary definition, referring to the caste or quality of authentic horses. So Müller-Wille and Reinberger, the ones I quoted before, the third one, quote, point to the erosion in the early 19th century of institutional barriers like those between naturalists and breeders as paving the way for the discourse of heredity. This is clearly visible in the work of Charles Darwin, and even earlier in that of his grandfather, physician Erasmus Darwin, who was also the grandfather of Francis Galton. So for the elder Darwin, analogies to animal breeding served not only to generate theories about the hereditary nature of human disease, but also suggested that human progress could be pursued biologically by learning the laws of nature and then exerting power over them. Indeed, for a number of physicians in the mid to late 18th century, comparisons to animal breeding pointed not only to the possibility of human improvement through control of reproductive practices, but also to its desirability and wisdom. So as animal breeding became increasingly elaborate and fashionable in 18th century England, it came to represent and perpetuate a particular modern view of the world. The English gentleman farmers who sought systematically to improve breeds of cattle and sheep during this period also established new institutions for the comparison and improvement of animal types, such as the country fair. A new world of thoroughbred horses sprung up, with a more simple admiration for fine animals being overlaid by systemization and officialized codes concerning the horse's genealogical blood and temperament. By the 1850s, the obsession with animal pedigree even led to a domestic poultry craze. Scarcely surprising then, writes Enrique de Cal, that this same period would see the high point of scientific and literary fascination with human types. Without attempting to make any extreme simplifying or totalizing claims, de Cal simply suggests that the animal breeding subculture was a sort of lower common denominator of biological information in the late 19th and early 20th century European societies, which was key to transmitting highbrow racist ideas to lowbrow audiences. in that it created a visual vocabulary and a living representation of race theory that was equally powerful on peasant farms or in urban settings. So too, in the early 20th century, in early 20th century American society, especially during the interwar period, their county and state fairs included not only exhibitions on animals and farming, but also those designed to popularize the ideas of the American Eugenics Society. Such exhibitions also featured the highly popular Fitter Families for Future Firesides contests. Eventually, eugenics exhibitions in the US found their way into national museums and larger world fairs as well. When we consider these ways in which the technologies of animal and plant breeding carved out and continue to shape the epistemic space of heredity, it becomes much more difficult to see the scientific thinking and discoveries that emerged within that space as merely neutral knowledge, whether that knowledge continues to be understood as true or, as in race science, has since been rejected as false. Beliefs in the power of human heredity to shape society and be shaped in service of society, engendered by success in animal breeding, are essential both to the race idea and to the support of any eugenic project. So how do liberal eugenics coerce? It's important for proponents of a new liberal eugenics that the project be understood as non-racial or non-racist. For them, this seems to mean that racial categorizations, as currently understood, should not be used in eugenic decisions. Thus far, I have used the notion of an epistemic space shared by race, heredity, and eugenics to trouble that understanding of racism. But it's also important for such proponents that their project be understood as non-coercive and respectful of individual liberty. So this is this concern I want to turn to. So Nicholas Rose describes how in advanced liberal democracies, the governing of individuals and the population is in large part achieved through means and in arenas that are deliberately designated as non-political. That is, through the regulated choices and aspirations to self-actualization and self-fulfillment of the people in the society. Insofar as national or social prosperity is thought to be achieved through individual prosperity, and insofar as individual prosperity is thought to require personal freedom, the state is believed to be taking a back seat, while individuals are not simply left, but rather exhorted to govern themselves. Thus, in liberal eugenics, reproductive and reprogenetic technologies must be chosen and used without state intervention so as to appear free from more obvious forms of power and domination. Yet these private interactions involve the exercise of great deals of power. To offer just 3 examples, Rose points out public distaste for state intervention does not necessarily lead to the banishment of authorities from our private lives. Rather, authority is accorded to formally autonomous expert authorities. For example, the parent who seeks to employ reprogenetic technologies as an investment in their child, or increasingly any parent at all, finds themselves very much dependent on the expertise of doctors or other fertility experts to carry out their project. Indeed, most parents rely on medical experts even to understand what is possible to desire and pursue in the field of reproduction through reprogenetics. Secondly, Michael Sandel argues that the deeper danger enhancements represent is a kind of hyper-agency. Far from simply allowing people to do as they like or freely pursue their individual projects, the availability of enhancements and biotechnological fixes appears as a bid for compliance, a way of answering a competitive society's demand to improve our performance and perfect our nature. With the idea that individuals control everything from socio-historical factors shaping one's life possibilities and outcomes fade into the background. And in the face of this dogmatic belief in individual responsibility, social solidarity is diminished. After all, if everyone is personally responsible for his or her own lot, there's little reason to respond with sympathy to those who are suffering or in need. And so similarly, Dorothy Roberts, third, argues that there are crucial similarities between reprogenetic technologies aimed at middle and upper class women, whose reproduction is generally encouraged, and those contraceptive technologies aimed at poor and non-white women. Both population control programs and genetic selection technologies, she suggests, reinforce biological explanations for social problems and place reproductive duties on women that shift responsibility for improving social conditions away from the state. Ultimately, insofar as they retain control over their reproductive decisions, women become gatekeepers of a new social order. My suggestion then is that eugenicists past and present share with each other and with race scientists and animal breeders a particular modern sensibility, a sensibility that sees the natural world as something that challenges humankind to intervene in natural processes in the name of industry, efficiency, productivity, and ultimately perfectibility. In this context, and today, eugenicists emerge as those believers in the power of science and technology who perceive clear and present dangers to humanity on the horizon and are thus driven to convince others that taking control of human nature, mental and physical, is an invaluable social good necessary to ensure human survival. So why does race still matter here? So what about the fact that most old eugenicists believed in biological race and most new ones do not? So even if we continue to debate the merits and dangers of genetic technology, should we stop debating them as racial issues? So by way of conclusion, I'll just offer 3 reasons to continue to consider race and racism when evaluating contemporary genetic technologies. First, we must recognize that the racist projects of the old eugenics did not simply disappear when the term fell out of favor after World War II. It was first replaced by ideas of family planning and population control, then ideas about overpopulation in the so-called third world, and the blaming of poverty among marginalized groups on too much production in the 80s and 90s. Second, it would be idealistic to point to the point of naivete to imagine that current racialized inequalities will have no effect on access to eugenic technologies or the benefits of eugenic projects. But also research in epigenetics suggests that environmental factors, both inside and outside the human body, have significant and ongoing impacts on gene expression. So this means that the proven disparities between racial and ethnic groups, both in mental and physical health, and in exposure to environmental hazards and toxins, could, based on their environments and discrimination, could translate directly into biogenetic disparities, even where everyone had access to the best genetic makeup biotechnology could achieve. Finally, even if all eugenic technologies were fully accessible and their use fully voluntary, the creation of 2 or more groups of human beings understood to have distinct biogenetic characteristics, capabilities, or capacities and incapacities runs the risk of creating all new races and accompanying racism. If anyone's watched The Expanse on Amazon Prime, it's a good example of this. And you should watch that. There will always be serious dangers to eugenic programs, especially where a reliance on biogenetic solutions to human or environmental problems leaves social solutions to those same problems untried, underappreciated, underfunded, or underdeveloped. If, however, such dangers truly pale in comparison to the global dangers eugenicists believe they can combat, they will need to pursue justice in their eugenic programs, not by denying issues of race and racism, but by confronting them head on, acknowledging both the complex racialized history of their drive to human mastery and its significant present day legacy. Let me unshare my screen. Okay. Stop share. There we go.
Great. Well, thank you both for 2 outstanding presentations, and you'll appreciate that I have a job cut out for myself in terms of offering any comments on both of those talks. I'm going to try, and I will very quickly turn it over to the audience if I fail to sort of be able to pull this together in ways that are engaging. So I want to start by saying, first of all, thank you for the 2 presentations. And I'm going to try to see if there are ways in which I can weave them together so that we can then open the conversation. So I think what we had at the beginning was a very helpful, accessible overview of the science of CRISPR technology, which is in fact a technology that has captured the moral imagination, if you will. And not so much in terms of people having great details about how the science does or doesn't work, but because a lot of effort, as was suggested, was put into using a metaphor that was quite accessible to the general public. We're talking about editing, and people understand that concept in terms of what you do with a text on your computer. You cut and you paste. I think we also, though, were treated to a very careful effort to call that into question. First of all, by making it clear that it's not as perfect as that sounds. And so Dr. Knoepfler took us through some of the things that could go wrong in terms of the examples of the off-target effects, the on-target effects, the unwanted deletions, basically making the point that we might have a particular project in mind, but that it's perhaps a little bit too much to think that we could execute that project without anything going wrong along the way. And I think that that's something that's really important for us to take into consideration, partly because we're going to be coming back to some of the comments offered by Professor Russell with respect to what is it that we thought the project was about in the first place, whether we actually achieve it or things go awry. So already you're seeing there's two levels at which you want to look at how things might go wrong. They might go wrong in terms of what you set out to think was the project, and any sorts of ways in which you might have cleansed that project from things that you perceive other people might think of as offensive. And that's aside from anything that might go technically wrong. The other thing that I think is really important for us to think about is what is ultimately the goal of this technology when we think about it in the context of manipulating human embryos. And I say that because very often the answer we are given, often I have to admit by the scientific community, is that this technology will be very important for eliminating genetic disease. But what we're also hearing from Professor Russell is that this technology would be one more way in which we would come into that reproductive sphere and actually try to do something that clearly has eugenic overtones. And so I think what's interesting is to be able to constantly hold these two presentations in our mind at the same time. So what are the goals? What are the objectives? Now, what's interesting is at one point, Dr. Knoepfler says, yes, but then I became aware that there were these alternatives. One of the alternatives, for example, might be something like pre-implantation genetic diagnosis. So the idea then being, we don't actually have to manipulate the DNA. We don't have to set ourselves up as the designers of the genetic material, but rather we would take a more modest role and simply be involved in making choices that have to do with selection between different bundles of genes that would be there in any case. But I'm assuming, and I stand to be corrected, that Professor Russell would say, well, actually, that's not getting at the issue at all. It doesn't really matter whether you think you're going to achieve your goal through selection or through design. What we're trying to make evident is what is the goal, and not just the stated overt goal, but what is in fact the socially constructed context within which you think you're making independent reproductive choices. And what are the biases that are built in that you may not even be closely attuned to because they've been normalized over a very long time, and a history that bears retelling. So I think that what we have here is already sort of two kinds of ways of asking questions about what are we trying to achieve, and relative to what we think we're trying to achieve, we're actually going to understand the problems of what it means to say something could go wrong. It also means that we're going to have different perspectives perhaps on what constitutes an alternative relative to the goal. But I think what both speakers are clearly saying to us in different ways from different perspectives is that there are significant ethical and social issues that need to be addressed, and that part of the work we have before us today is actually surfacing those in a way that they can be heard, that they can be engaged with and responded to. And I think for me, one of the things that becomes really important in the context of these two talks is there was a statement made by Dr. Knoepfler where he says the cultural differences make consensus very difficult. I'm paraphrasing. I hope I got that right. I stand to be corrected. But I want to say one of the things that I think is really important is maybe we're giving up too easily on that. And I'm going to put this idea out there because I'm hoping that maybe both of the speakers would like to engage with this idea. Is it true that we are not in a position to work towards any kind of consensus building with respect to any kind of technology that we might use in pursuit of any kind of goal. And you'll notice that I've sort of tried to state that sentence in a way that tries to leave it open. And I'm sure if I had a few minutes to write it down, I would, recognize that I haven't made it as open-ended as it needs to be. But let's just play with that idea. It has become very easy in the contemporary world to focus on our differences, and they are many, and they are important, and they are sometimes difficult to overcome. But I want to hold out some hope for the possibility of consensus around critical issues like, what might this technology be good for? What are the harms of this technology? Why might we not want to use this technology? And in here, you can put any kind of reproductive technology, genetic or reproductive technology. I don't mean to now limit it just to CRISPR technology. And I think what I'm wanting to say is that if we understand the project of consensus building not being about unanimity, okay, so I'm actually not so naive as to believe that we could get 8 billion people in the world to agree about anything. But I think there's a big difference between unanimity and unity. And I think one of the things that we haven't explored is, what is it that we have in common as opposed to where is it that we're different? And I think that in part, as we surface some of the differences, we may in fact be able to turn them around into commonalities. And I think that that can be the case, even with such politically difficult and complicated issues like race and racism. So what do I mean by that? I think that there are ways in which what we need to have is space to have conversations where people with different expertise, different knowledge, different lived experience get to contribute to a conversation, get to call into questions the assumptions that are taken as normal. And in that, the goal is to get to a point where, as I've said, you have unity, not unanimity. What do I mean by that? It means that you're working towards an understanding, a consensus that isn't going to be achieved at when we all agree on an answer to a question, but rather that we all agree that our views have been heard. So for example, I think that the kinds of really complicated and important views that Professor Russell is unpacking for us when she's saying things like, racism didn't go away and the eugenic project hasn't gone away, it's just been morphed, it's been sanitized, it's been reframed in such a way that it's easy for somebody to speak about certain choices as private reproductive goals, in no way in pursuit of any kind of a eugenic project. And yet, if we really listen to what's being said, there's an opportunity for us to open our perspective on that issue and to begin to see it and understand it differently. Well, that's what I think is really important. And that's why I think we shouldn't give up on the idea of consensus, because we have many things in common. And the main thing we have in common is a desire for survival on this planet. And if you want to think about that in Darwinian terms, and then you want to worry about what that might mean in terms of survival of the fittest, then we have political goals to make sure that we're actually redistributing the cultural, social, economic capital such that the survival of the fittest now means most of us as opposed to the 1%. So we do have the prospect of coming up with common goals and objectives that might have us see certain uses of certain technologies as worthy of pursuit. At the same time, it could go in a different direction when we're persuaded there's in fact no way, quite frankly, given our limitations, that we would ever be able to use this technology in a way that would not exacerbate important harms in terms of risks of discrimination, marginalization, et cetera. But we might be able to come to that conclusion together if we were to be open to the possibility that there are different ways of understanding the world in which we live. And I think that, in part, is a major contribution that Professor Russell is trying to make here. And I think it can build on what I think I heard Dr. Knoepfler say with respect to that divide between humility and hubris. you know, that we're these scientists and we've got this great science and we can do these wonderful things. And then, you know, being reminded, yeah, but these are all the things that can go wrong. And then having somebody with a non-science perspective on what it means to say, these are all the things that can go wrong, having a space now that's been opened up to say, and I have something to add to that story about everything that could go wrong. Now, one of the things I want to say for me that's really important in advocating for this consensus building is obviously it's somewhat self-interested because it's a view that I hold and I advance. But I do want to say at the end of the day, even if I'm wrong and it's not possible and we fail, it would be a better world for having tried. It would be a better world for the scientific community and the humanists to sit down together and to try to say, this is where I understand the problems. And no, they're not technical, they're not scientific, they're social and they're cultural. Now, one of the things that I want to say, you know, that I thought was really striking in this is I had the pleasure of reading, you know, Dr. Russell's recent book and preparing for today's conversation. And I was struck by very early on, she refers to work by Neil Postman. And, you know, he says that when we look at technology, one of the first things we should do is ask ourselves the question, what is the problem for which this technology is the solution? And for me, I keep coming back to that question in my own work, insofar as I think we have moved so far away from that question that too often it's a technology searching for a problem. And I think that that's where, if we could even have a conversation about what that means. So for example, if we go back to the, you know, early information that we were given about CRISPR, it was a technology that was in part developed to deal with a specific problem in yogurt. But there was a problem that people were trying to solve. And now that we have this technology, it's just really cool science. And aren't there things that we could do with this really cool science? And oh my goodness, we could help people who are suffering with a genetic disease. And then the next thing you know, one answer to the question becomes, well, another desirable outcome would be not actually to just look at disease, but then to look at difference. And we could make perfect humans. And then we, I think, circle back to a point that Professor Russell is making, which is, well, what becomes the reference point for the perfect human? And I think, you know, she says quite clearly that the reference point is probably going to be a particular understanding of the dominant community right now, at least in the Western world, which quite frankly is people who are part of what would be seen as a white racial or ethnic group. And I say that because so much of that has now become the norm, that that's the reference point. And I can even say that with respect to a new word that's in, I think I can't remember if it was the Webster or the Oxford Dictionary, but I did look it up to make sure. And, you know, biopic is now in there. And, you know, I want to say why. Why do we have a term that formalizes people of color when that can only make sense relative to what? The absence of color? Who are the people walking around with no color? If these are the people of color, then I know what that means, right? And so I think that it's in that kind of context that we're being invited to problematize the use of this technology, to understand how quickly it can move from one kind of stated goal to another goal, which ultimately maybe wasn't the problem that the technology was supposed to be a solution for, but which after the fact can be reframed. And I certainly think that we have seen a little bit of that with the CRISPR technology. I just want to end with one comment that I think is really important, that I do think is one of the many points that Professor Russell was trying to make with respect to the Eugenic project when she says it never really went away. I think that one of the ways, at least that I always understand that, is in terms of there kind of being this agreement that something that would be imposed on people in terms of controlling their reproductive choices by the state would be wrong, but that if individual people were making independent reproductive choices, that if they happened to independently make a choice that furthered a eugenic goal, we wouldn't thereby call it eugenics, because somehow eugenics has this pejorative connotation attached to the exercise of power by the state. But I think part of what we're being told and reminded is that that doesn't make the concept or the idea of eugenics go away, and that we always have used that concept in a way that has meant to improve a particular understanding of what it means to be a perfect human. So with those questions and remarks, an attempt to try to pull those two disparate presentations together, I invite questions and of course corrections from the two speakers if I have it wrong.
Well, thank you very much. I want to thank all of our speakers, Professor Knoepfler, Professor Russell, and Professor Baylis, for their illuminating talks. And so now we're open to questions from the audience. And so I'm going to start out with a rather broad question, and I'm going to direct it to whomever would like to answer it. And the question is, who gets to decide? And I think this starts thinking about what Professor Knoepfler told us about the list of 10 genes that a famous geneticist thinks might be good targets for CRISPRing in people. And it carries over into the idea of eugenics. And I guess a piece of this is thinking about whether we have state mandated policies, whether we have choices being made by individuals, and in our increasingly transnational world, how do these things dovetail? So this is a very open question, and I'm just going to throw it out there and let everybody who's interested respond to it, and then we'll move on to the next questions.
I can start with a couple comments. So it's been interesting for me just throughout my career in science to note that there is sort of a, I don't know the best term for this, but almost like a scientific elite in a way that I've seen throughout. I first started as a technician and then a student and a postdoc and now a professor. There's sort of a celebrity class almost, if you will, of scientists who have, at least as one can think of scientists contributing to the dialogue and decision making, there's sort of that group of elite scientists who have the most power to influence decision makers. We see that through groups like the National Academy of Sciences, selecting maybe perhaps the most famous scientists to serve on an advisory panel on germline gene editing, people who give talks at meetings about heritable germline gene editing. Who selects those people to give talks? And you know, so it is interesting to think about if there are contributions of scientists, and by that I mean people who are doing genetics work, let's say, or something like that, working with CRISPR. You know, who amongst the scientists really should be the decision makers or the contributors to the dialogue? Should it be the people who are the ones who invented a certain technology? You know, is there some other way we could evaluate who perhaps has the wisdom, you know, not necessarily just brain power to invent a technology or something like that, but the wisdom to contribute as a scientist to the dialogue? So I do think amongst the scientists, there's definitely going to be influencers who help make the decisions that are made. But I guess I wonder, what's the best way? What's the way with the most wisdom for that to happen? And I don't know. I don't have the best answer to that.
Well, I had the pleasure of reading Dr. Baylis's book in preparation for this and an interview we're going to do tomorrow. And yeah, I feel like that book basically it tackles this question: who gets to decide? You know, it's a whole nice, actually very accessible book on who gets to decide. But one of the things that worries me is a sort of like non-decision, right? So it like if global capitalism decides, because global capitalism is not like an agent, but it has a lot of, you know, it does a lot of things in the world, right? So I worry that people won't decide per se at any moment or through any processes of deliberation or consensus making, and that, you know, the sort of, the forces of global capitalism will just kind of lead in sort of incremental ways to things just kind of going forward, right, without sort of that moment for a collective decision. So I mean, so for me, it's like, let's get someone to actually, in a transparent way, talk about deciding, right? To, you know, even, you know, so that we don't just go forward without, it doesn't just sort of happen.
Well, well, I think that one of the things that I do believe, and I, want to say in response to the first, answer offered by Dr. Knoepfler, that I don't really want it to just be broadened from the elite scientists to the scientific community. Because for me, that's just not broad enough. And I've said repeatedly, the decisions that are before us are just far too important to leave to the scientific community. And that's the move that I then make to say it's also too important to leave even to our elected officials, for people that live in democratic countries, for many of the reasons that actually tie into some of the limitations with respect to capitalism and the ways in which I think capitalism has completely distorted democracy and how that works in a contemporary context. I think for me, the project is really to try to make people be less complacent by understanding there are decisions to be made. And I think that for a long time, we haven't felt like we had any opportunities to have any influence on really important decisions. And in cases where we have tried, we have not been listened to. And so I actually think that this isn't even just about CRISPR, it's about science. And quite frankly, for a very long time, science has operated in a way that it kind of has a privileged status. And the idea is that society will reap all these benefits. So you just let us do our thing, and then you will have the benefits of our science. And I think that we're at a turning point where it's important for us to have allies within the scientific community who will help to make the science accessible, but that in many cases, it's not the great details of the science that people need to understand. It's the questions that the science raises for us, about which we all have opinions. And I think that's why, at least in my remarks, one of the things I keep wanting to emphasize is the importance of a certain amount of humility on the part of all of us. And that humility means we don't have an answer, but maybe somebody else in the room does. And then what becomes really important is who gets into the room, which is something that was alluded to. And I think that there are ways in which those spaces are gendered. Those spaces are definitely not welcoming to people who don't have a history of working within those ways of interaction. And so we have a lot of work to do. But I think people like Professor Russell, just not accepting, claims that people might otherwise be accepting of, so just saying, look, this is a eugenic project. And people are going to say no. And then you say, well, if you're willing to listen, I can explain to you why it maybe is. So I think it's that openness that we have to start really cultivating if we're going to deal with many problems and not just this technology. I mean, I really think it's many reproductive technologies, but it's also, technologies that have to do with efforts to address problems of pollution, of food insecurity, of climate change, et cetera. And it cannot just be those who understand the science who will determine the way in which the science can meet our goals and objectives.
So the next question, I think, is going to be addressed particularly to Professor Russell, but I hope everyone else feels free to chime in. So race is something we hear a lot about these days in the media all the time. We've brought it up here. And yet race is something that does not have a genetic basis. And so I think maybe it would be nice if we could have more of an explanation of what that means.
Oh my goodness. Yeah, I mean, I talk about this all the time, but I don't want to take all the time talking about it now. So race is not, what we talk about as race and the idea of race as we have it now, complex as though it may be and historically situated as though it may be, does not, there's nothing that in genetics that maps onto that historical idea of race, right? So even if there is some grouping of people that science could come up with that wasn't purely arbitrary and that's questionable, there's always lines to be drawn, that more scientific grouping wouldn't be race as we have it now, because it has just like too much of a history, right? But yeah, so I mean, for me, race is an idea. It's a concept. It's real, like money is real, and both are probably equally dangerous. But it's, I mean, it's this thing that is created. It's a social construction, but it operates, right? And you can't get along. The world will not behave, society around you will not behave in ways that make sense to you unless you know what money and race are and how they are thought about in your particular context, right? It's otherwise just everything's not going to make the right kind of sense. You're not going to understand why people are afraid of some people and not others, or why people look the way they do in mass incarceration and stuff like that, right? I mean, it's all you have to know kind of all this stuff for everything to actually make sense. So it's a social construction. It has a form of reality that social constructions have. People act as if it's real in a lot of different ways. That doesn't have to mean it's biological. But it does have, you do get bodily effects of being racialized in a particular way. This is what we do in the humanities. We add eyes to everything. So we don't want to believe in sort of race as a biological construct. We can talk about people and their bodies being racialized, right? Being understood as belonging to a particular race, and thereby being associated with particular ideas about those bodies, right? So, and this can be in a real sort of health way, right? And there can be actual changes in the body that are related to the experience of the body being racialized, right? The experience of chronic discrimination can create forms of chronic stress, right? That can contribute to a lot of health disparities, infant mortality for Black women, or low birth weight children among Black women. So there's a lot of different ways, and then as I briefly mentioned, there's sort of environmental injustice and environmental racism in the way that people might lack access to healthy foods, or might be exposed to lots of toxins in their workplace, in their home, those kinds of things. So there's all these kinds of ways in which the fact that you're seen as a member of a particular race through forms of discrimination and segregation and other kinds of marginalization can actually affect the body. So I think there's a real effect on our embodiment by being racialized, but that's not to say that race is a biological entity found in our genes.
If I can just add to that, I think one of the things for me that's really important, a facet of what is really important in terms of understanding how race works, at least how I think about it, is it's a cluster of biases. And we ultimately teach those to people. And I think, I first became aware of that when I realized I hadn't been taught properly, because I couldn't discern things that were clear to everybody else. So for example, a trivial thing, you go into the room and you ask, and you're talking to somebody and they say, oh yes, well, the Jewish man. And you say, well, no, how would I know that? Was there somebody walking around with a sign that said, I'm a Jewish man? Like, I don't know who that is. Being in a conversation and we were talking about Indigenous people, and he says, oh yes, but they're not like all the other ones. What are all the other ones like, right? I mean, there are all these ways in which we actually start creating a set of norms around, as I said, a cluster of ideas that somehow take on a life of their own and a meaning of their own. But what matters is that people know what those ideas are, however it is that they're clustered. And so I think that what happens is, just as people internalize it in order to be able to identify the other, the person who is othered also internalizes that information. And I think that those are the ways in which that those ideas move, and they can do great harm. But actually, I think they also do harm to the people who are trained to know how to use those concepts.
Thank you. So the next question is directed at Professor Knoepfler. So CRISPR and other genetic modification technologies are routinely used in laboratories around the world, many, many laboratories. And they're used on research animals, mostly with the goal of understanding how some process normally works, and then what might go awry in that process if there's a disease situation. And so how do you see that work intersecting with the idea of human genetic modification? It's like there's a little bit of a disconnect there.
Yeah, I think there there can seem to be a disconnect, but for instance, at my home institution, UC Davis, we actually started as the agricultural school for UC Berkeley. So even to this day, 100+ years later, we have a major agricultural research program going on, and there's lots of CRISPR work being done in that context. And there actually is a connection there, even just in one institution, between researchers doing different kind of work, but perhaps with shared technology like CRISPR. So I know not only here, but at other institutions, a lot of the work being done in, in cells, excuse me, and in animals, whether it's for agricultural purposes or research purposes, those researchers working with the animals, they keep in mind, they're thinking about the human context as well. And I gave the example of the CRISPRing of the myostatin gene to create larger muscled animals. And part of, I think, the goal there was to learn about muscle development. There's potentially an agricultural goal to make animals that have more meat per animal. Maybe that also feeds into an economic kind of goal potentially. But that research it sort of ends up being within the context also of the human condition as well, because, you know, first of all, like in the case with George Church, on his list was that same gene myostatin to potentially edit in people with the goal to make a change in people. And so same gene, you know, same technology, CRISPR. In one context, we're talking about pigs. In another context, we're talking about people. So at least from my experience as a scientist, you know, doing biology and genetics, I often see that kind of thing, like we were talking about myostatin, but with other genes as well. And so there are, I think, many parallels and connections between the work in animals and what goes on in people. And certainly, for instance, with the other gene CCR5 related to HIV infection and other kinds of viral infections, that work also is ongoing in animals, it's ongoing in people. So for me, I see the two as kind of parallel and interconnected.
So here's another question. I just read Lord of the Rings and was struck by the obvious influence of eugenics in its understanding of races in the books. So men versus dwarves versus elves versus orcs, etc. So how do you think eugenics, or the modern day descendant of eugenics, or CRISPR and all of the things that we've talked about will continue to pop up in our popular culture? And I'm going to open this to everyone.
Well, I can make a few comments. I definitely have seen it in pop culture quite a bit, usually in movies that are sort of trend towards being like adventure kind of movies, spy kind of movies. And they generally, there's a couple of themes there. One is the idea of enhancement, like enhancing the human condition somehow to make like a better spy, a better soldier, something like that. And then also, as I briefly mentioned earlier, there's also sort of the side to it of what can go wrong, creating some monstrous kind of creatures with CRISPR by accident, something like that. So I think it's definitely penetrated in pop culture. I've talked to many, you know, like family and friends who are not scientists, not physicians, anything like that, and they're very aware of CRISPR. They know the basics about it. And, you know, I sometimes give them kind of an earful, like some of the things I said in my talk today about, you know, the complexities that are really important and you know, the ways it can sometimes not work. But I'd expect that we'll see more pop culture kinds of things, TV shows, movies, books, streaming things, you know, streaming services, productions that weave CRISPR in there, and larger themes related to genetics and the human condition.
I haven't read the book, so I just can't say anything.
Oh yeah, there was the question of the Lord of the Rings. Yeah, I've read them, but it didn't really occur to me, and maybe, I don't know why, but it did occur to me there were clear differences in the characters, but I didn't think of it so much in the context of like a eugenics kind of way of thinking.
But I think that would have been the author's context in early 20th century England. So, I mean, I think that probably what you're seeing is this pervasiveness of the idea of eugenics as a sort of transnational modernist philosophy, you know, in that book, or those books, all the books.
So there's a question here, I think directed at Professor Russell, but again, I invite everyone to address this. So eugenic ideas have been embedded in law and policy governing reproduction in the past, and so have efforts to counter eugenics and even genocidal efforts. For example, the Indian Child Welfare Act. So what forms of regulation do you think might be appropriate as we move forward?
Yeah, that's a big question. I mean, absolutely, it's absolutely correct to state that you know, you have policies that both incorporate the idea of I mean, if as I sort of argue, if we think about these kinds of things, or if one aspect of eugenics is crucially this sort of idea of these biological solutions to social problems, or to fixing social problems through controlling reproduction, I think, yes, we do see this, you know, and this is the sort of criticism that I didn't go into deeply when I talked about the bioethicists who said that they thought reproductive freedoms were well embedded, right? You know, so we have, you know, we have the 90s example of, you know, linking welfare and welfare benefits to not having more children, or to having long-acting reversible contraceptives, some of which were fairly dangerous. You know, implanted, Norplant was one of the big ones. You know, so there's a lot of ways in which people have tried, you know, in the U.S. and in the U.S. even acting globally has, you know, tied sort of different types of foreign aid with different types of sort of family planning programs and things abroad. So yeah, there's tons of stuff, but I think it's hard to know kind of how that will happen in the future. I mean, you know, I think, yeah, it's hard to speculate exactly how these things will kind of get taken up, and some of the more subtle ways that they might appear in law. So I'm not sure, but I mean, I suppose one thing, thinking about Dr. Baylis's book too, that I worry about is the sort of idea of genetic privacy, which we have increasing laws around, giving the impression that one owns one's own genome, and therefore taking away from part of what Dr. Baylis argues in her book, this idea that the human genome belongs to all of humanity in this way, right? So I think we might even sort of see some of this sort of individualization and privatization of genetics in some policies right now that are well-meaning, in terms of, you know, genetic privacy policies are well-meaning, but I do wonder if they shift things away from the sort of common inheritance that that Dr. Baylis talks about in terms of how we should think about the human genome.
Yeah, if I can, I'd like to offer two comments. I think when you think about the ways in which science and the law, or specifically assisted reproductive technologies, or just other genetic technologies, intersect with the law, I think we need to always have two things in mind. First of all, the ability and the will to identify laws that are unacceptable because they are ultimately discriminatory. And, you know, I think there's this sense that in the modern world, we don't have those kinds of laws, but we do. And the very first part of the project is to see what are the laws that are on the books that need to be removed, number one. But number two, we then have to go further and not look for that which is overt, but look for that which is indirect. And so as an example, I know that in the United States, you don't have a government-funded healthcare system, but pretty much every other country in the world does. And one of the things that's really interesting is to look at which countries will pay under their healthcare system for assisted reproductive technology, which can be very expensive, but will not pay for adoption. So they will pay for a technological response to what you might, as an individual, identify as a problem, a desire, a wish. You would like to have a family, but you might be quite happy with adoption as the strategy that you would like to use to build your family. And then it becomes financially not possible because adoption actually is a very expensive exercise, as is the use of certain reproductive technologies. But people would not necessarily see that up front as any kind of problematic policy. But I think it's always a problem if the state privileges one kind of way of responding to a problem and nominally choosing technology when there may be other ways of responding to the same perceived problem. Now, I'm not saying that it always has to go in that direction, but I'm just saying, or what I'm trying to communicate is there's two things you need to look for: things that are clearly overtly discriminatory and things that actually end up entrenching values that are potentially, let's say, problematic and worthy of being questioned, because otherwise it just gets normalized. And I think for me, that overlaps with a kind of deference that we have to technology, which brings me back to the question, what's the problem this technology was supposed to help us solve? And I think too often we don't actually start with, we have a problem, let's see what we can do to fix it, because sometimes you would actually use existing technology, right? Because it would be the right thing, the most effective thing to do for a particular kind of challenge. And I'm reminded, way back when I received in the mail a report telling me, about the top 10, biotech for the developing world. And I remember screaming, who wants to know about the top 10 biotech needs of the developing world? I want to know about the top 10 problems. And then we can figure out what solutions we might be able to offer to those problems. The first question is in the service of science. I got a lot of cool technology. What are the top 10 techs I can take somewhere, as opposed to I'm part of a community and we're experiencing problems and we can't do everything. So we have to prioritize our problems so that we can try to solve them. And I think there are questions of orientation that we can look for in the ways in which we regulate ourselves overtly with law and regulate ourselves indirectly with capital. Capital meaning money.
I think another interesting question, and I don't have a very good answer to this related to law and the kinds of topics we've been talking about, is whether or not there should be a law or some kinds of regulations regulating heritable use of CRISPR in people. So this would be kind of looking to the future, and I know this has been discussed in the scientific community and other communities, but or is something, so far there's sort of been this slapdash approach where the FDA is prohibited from considering applications for new drug kind of efforts of biotech companies related to heritable use of CRISPR, which sort of is like a partial block from the biotechnology kind of sphere in terms of not allowing heritable use of CRISPR to sort of be advanced from a biotechnology company perspective. But I think there's sort of this fear in the scientific community that there will be a law passed that in some way sort of restricts or constrains use of CRISPR in human embryos in more of a dedicated formal kind of sense, rather than just sort of these improvised ad hoc kind of approaches to restrict the FDA. So, and I don't have a good answer, because as a scientist, I don't necessarily like the idea of a law telling scientists what they can or cannot do, but obviously there are laws like that. And there are other countries besides the US, and there actually are other countries that have passed laws related to CRISPR. So, to me, as I hear about laws, I think about, possible pluses and minuses of going down that route.
Well, if I can, I would offer a couple of comments. One of them is to say that I do think the view that we don't like laws is kind of widespread amongst the scientific community, or if we don't like laws, we don't like rules. unless we've developed them ourselves, which is this idea of professional self-regulation. And I'm certainly amongst those that are willing to question, by virtue of what scientists get to do self-regulation, when in fact they benefit tremendously from the social cultural context in which they're able to work, and from often resources made available to them through tax dollars to fund their research. So that's the first thing I think we need to be willing to interrogate and have a conversation about. So as I said, I'm quite aware that many in the scientific community think professional self-regulation is the way to go. And the standard reasons given for that are things like the science is moving too fast. We can't put in these laws because then we won't be able to change them. The idea, of course, being that we need to change them to kind of keep up with the science. And that's actually a frame of reference that's worth interrogating. Why is it that we think the law lags behind science, or ethics lags behind science? They're actually two completely different frames of reference, and it doesn't make any sense to assume that what science is doing is right and the rest of us need to catch up to science. Maybe science is, racing ahead and creating problems. So that's one thing I wanted to say. But the other thing I wanted to say, which is really important, is when I started doing work in this area, I was amongst the people that called for a moratorium on the use of this science, with the explicit goal of saying we need to be able to have a conversation. And if gene edited babies are just going to be popping up in different countries at different times, how can we meaningfully have a conversation about whether or not to use this technology for what end? Because de facto that decision would have been made by individual scientists in different countries, just barreling along and doing what they wanted to do. But in that context, as I said, I participated in the call for a moratorium, but I did that without having background knowledge as to what was the global policy landscape. And I and colleagues have since done that research and, quite frankly, came up with surprising findings. We looked at 193 countries in the world, plus the Vatican and the state of Palestine. And out of those, we were able to find policy information documents for 106 countries. So that's actually quite a number of them. We found information about 196 countries. We found actual policy documents for 96 countries. And the most interesting thing about those policy documents, when you look at them, is that 75 out of the 96 have a prohibition on this. And so I now ask people to think about, well, if you were objecting to a moratorium, who is it that you didn't want to have any prohibitions? Because we already know that half the world, if they've actually bothered to think about this, and I should reframe that. I should not say bothered to think about it. That makes it seem like as if, they didn't have that as a priority. There are lots of reasons why lots of countries should not have this as a priority. They have all kinds of other things to worry about and legislate about, et cetera. But of those who did actually have documentation that was relevant, they actually have a prohibition. And we did not find a single country that explicitly permits it. And so when people say no to a moratorium, they're actually saying, oh my gosh, well, we have all these countries that have already prohibited it. We better make sure that those who haven't done anything don't have a moratorium. And that then takes us back to questions around, travel, either between the scientific community or with the transport of tissues, which we've seen happen to try to evade places that do have regulations. And that's why I think, we do really have to try to find a way to have a conversation, because the whole point isn't to say, well, we've got prohibitions here, so let's keep other places open so that we can move around the world. Science is global. And the problems that science creates are global, but the problems that science could help fix are also global. And I don't think that we have the right perspective yet on what it means to do good science.
So I think CRISPR is a great example of a technology that was discovered not because it has a purpose. It was discovered, as Professor Knoepfler told us, in basic research, trying to understand how bacteria can keep themselves from falling prey to viruses. It was a very basic piece of research. And then, we've had this DNA revolution a number of decades ago, and people who work in that area suddenly realized that there was a great deal of potential for co-opting this system that already existed and modifying it in a variety of ways so that it could be used in animals, plants, and potentially in humans. And it happened very quickly. It's just a few years. And I think that, as Professor Baylis has pointed out, we don't necessarily have the policy or the wherewithal to have policies in place as things are evolving, and we don't necessarily have conversations between the scientific community, policymakers, ethicists, lawyers, and the global world population at large to figure out how these things should be, I'm not going to say regulated, managed, how we should put it into some sort of context to think about how we want to move forward or not move forward. And, you know, CRISPR is not the only thing that's been discovered in this way, and it is probably not the only thing that will be discovered in this way. We don't know what revolution is around the corner that will be discovered. That's the nature of scientific discovery. So I'm going to put this out to all of you. Who do we need to have at the table, and how do we arrange that table in order to think about these issues and think about how we want to deal with them in national sense, in international sense, in a global sense, in a way that is as positive as possible for the planet and all of its inhabitants, and yet doesn't prevent us necessarily from having the next big breakthrough that might be really fantastic for something. We don't know what that is. So I know that that's completely open-ended, but I will let people comment as.
I'll make one quick comment, and that is, having attended the 2015 National Academy of Sciences, like the first meeting about CRISPR use, just as an observer, I didn't speak or anything. And then I watched one of the more recent ones, I think it was in Hong Kong. Just as an observation, I would say it's maybe 80, 90% the scientists who so far have been at the table. They're the ones who have a voice. They're the ones given a voice. They're invited to speak. And I think that's not gonna do it, right? Moving forward, I think that's not the way we should go. I think we need a much more diverse group of stakeholders at the table, and not just, you know, obviously I'm a scientist myself, but I think having almost all scientists be at the table is not the way to go. And by scientists, I mostly mean like biologists and geneticists. And
I mean, Dr. Baylis has literally written the book on this, so and it was very good. So I would allow her some time to answer this.
Well, I think that the main thing that I want to say is that we need to respect different ways of knowing, and we need to understand that people have different kinds of views and values about what's important. And that if you actually took time to talk to people, you might be surprised about some of that. I know that I learn constantly. And I think that one of the things we don't do is assume from the starting point that people who have something to contribute get a space at the table. So in a way, that's partly how the question was put, but in my book and in some other work I do, I actually use the metaphor of the kitchen. And I say, you know, what if all these really important conversations had to happen in the kitchen? And, you know, part of that is to try to communicate that, you know what, we all have to eat. So it's a reasonable space for us to go into. Kitchens and eating is a very social thing. It's something we do to welcome people. We share food with them. The rules of engagement are really quite different. I don't have to know how an agenda works, and I don't have to, wear a suit or a white lab coat or do any of the other kinds of performative activities that give me stature in the conversation. And I actually at one point even bust out and say, you know what, maybe what we really need is a picnic. And that was in part even to recognize that there are parts of the world in which, kitchens don't function as we might think kitchens function, and you live outside and you eat outside. And it really is to say, I think there's a lot of work to be done in space, in terms of how we organize ourselves. And I think that I've tried to use there a different metaphor, which is the metaphor of architect, and that it's really important to design spaces in which people can contribute. And there are people who are well-meaning, but honestly, there are people who kind of say, but the door's open. I don't understand why nobody's coming. And it's like, yeah, well, most people know what happens once they step over the sill, and it's just not a friendly place. So why would anybody come? And if you're really not going to listen, but all you want to do is check off the box, and that says, I did some public education. Maybe I don't want to support you in your project of being able to say you did some public education when you didn't actually really want to hear. So, if you think about what I'm trying to say in this most radical space, I'm actually trying to say we need to share power. And right now, the people who have power aren't so keen on that project. And that's kind of understandable. If you have a lot of power, you don't want to give it up. But that does need to happen.
Well, I think we are at the end of our conversation. I want to thank Professor Knoepfler, Professor Russell, and Professor Baylis. I think this has been very informative, very exciting, a great deal of food for thought, a great many ways that people who aren't scientists could become involved in thinking about the science, and people who are scientists could become involved in thinking about ethical issues and other impacts. And I look forward to a time when we can have more of these conversations. I want to thank the audience, and now I'm going to turn this back over to Professor Herman.
Thank you so much to all of you. I'd like to echo Professor Eisen's thanks to all of our panelists and to members of the audience. I don't usually think of the Wayne Moore Center as a kitchen, but as somebody who loves to cook and eat, and of course share food in person, something I know that a lot of us are really missing. I think it's a wonderful way of thinking about the project that you all have been talking about for the last hour and 50 minutes or so. It's certainly the project of thinking about genetic technologies, advancing the goal of public engagement, inclusive democracy and justice. I draw those words from the mission statement of the Wayne Moore Center, but I know from the conversation that you have had and the questions that we got that those are goals that are important to all of you. They include scientists, citizens, humanists, scholars, members of all kinds of communities, people making reproductive and other health decisions. On behalf of the Wayne Moore Center, I'd just like to thank you and all of you in the audience one last time. Please do join us if you would care to do that for future events in this series. And also please be aware that recordings of a number of our past public events in the science policy and the public series are available both on the Wayne Moore Center's website and on our YouTube channel. Thanks again, goodbye, and stay healthy.
Thank you.